I think we haven't been able to find a biomarker for these ADCs yet, with the exception of Trastuzumab deruxtecan. Right now, it is exclusively used outside of a clinical trial for patients with HER2 mutations. But hopefully patients with HER2 overexpression might benefit from this drug as well. And I'm talking about non-small cell lung cancers, obviously. There's the whole story about breast cancers. For the TROP2 ADCs, TROP2 is so ubiquitously expressed in non-small cell lung cancers that it really doesn't serve as a good biomarker. So as of yet, this drug is being used in all comers. And we also saw some very interesting data of Patritumab deruxtecan. So that's an ADC against HER3. And at this year's World Lung, we had new data presented on the activity of this drug in patients with EGFR mutations that have failed osimertinib, and the response rates were around 30%. So this is another biomarker. I mean, it's not for the use of the antibody because this antibody has been shown to be active against all mechanisms of resistance, but probably the first approval for this antibody is going to come specifically in patients with EGFR mutations. And I think the promising combinations are definitely going to be with immunotherapy. And there has been some combinations with the ADCs with targeted agents for specific subpopulations of patients with actionable genomic alterations. So there are combinations, for example, of MET-ADCs with EGFR inhibitors and so on.